Genomic and non-genomic effects of hormones. The actions of several hormones listed previously which act on receptors within the cell or nucleus causing gene transcription, target gene expression, and ultimately cellular responses are referred to as genomic effects of a hormone. And more recently, novel methods of signal transduction through steroid hormone receptors that do not involve gene transcription have been identified for several hormones. These non-genomic effects do not depend on gene transcription or protein synthesis. The novel mechanism involves steroid-induced modulation of cytoplasmic or cell membrane-bound regulatory proteins. The hormones that have both genomic and non-genomic effects include estrogens, progestin, aldosterone, and androgens. The characteristics of non-genomic effects include the following. The actions are usually too rapid, seconds to minutes, to involve any changes in mRNA and protein synthesis. These actions can be observed even in highly specialized cells that do not accomplish mRNA and protein synthesis, for example, spermatozoa. The non-genomic effects can be elicited even by steroids complete with high molecular weight substances that do not pass across the plasma membrane and do not enter into the cell. So, non-genomic effects are not blocked by inhibitors of mRNA or protein synthesis. Non-genomic effects are not blocked by antagonists of the classic genomic steroid receptors. Then, second messengers mechanisms for mediating intracellular hormonal functions. Definition. Second messengers are molecules or substances that help communicate signals from the cell surface to specific target site within the cell, either in the cytoplasm or nucleus. Second messengers help in significantly amplifying the strength of the signals. There are several types of second messengers, and these are listed and explained. We note earlier that one of the means by which hormones exert intracellular actions is to stimulate formation of the second messenger CAMP inside the cell membrane. The CAMP then causes subsequent intracellular effects of the hormone. Thus, the only direct effect that the hormone has on the cell is to activate a single type of membrane receptor. The second messenger does the rest. So CAMP is not the only second messenger used by the different hormones. Two other especially important ones like calcium ions and associated calmodulin. And second one, products of membrane phospholipid breakdown. Adenylyl cyclase CAMP second messenger system. Binding of the enzymes with the receptor allows coupling of the receptor to a G protein. If the G protein stimulates the adenylyl cyclase CAMP system, it is called a G5 protein, GS protein, denoting a stimulatory G protein. Stimulation of adenylyl cyclase, a membrane-bound enzyme, by the GS protein then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate into CAMP inside the cell. This then activates CAMP-dependent protein kinase, which phosphorylates specific cell proteins, triggering biochemical reactions that ultimately lead to the cell's response to the hormone. When CAMP is found inside the cell, it usually activates a cascade of enzymes. Activated. In this way, even the slightest amount of hormone acting on the cell surface can initiate a powerful cascading activating force for the entire cell. If binding of the hormone to its receptors is coupled to an inhibitory G protein denoted by GI protein, adenylyl cyclase will be inhibited, reducing formation of CAMP and ultimately leading to an inhibitory action in the cell. The next, cell membrane phospholipid second messenger system. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C, which I have heard about this, attached to the inside projections of the receptors. This enzyme catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidylinositol biphosphate or PAP2, into two different secondary messenger products, inositol triphosphate IP3 and diacylglycerol DAG VAG. The IP3 mobilizes calcium ions from mitochondria and the endoplasmic reticulum, and the calcium ions then have their own second messenger effects such as smooth muscle contraction and changes in cell secretion. JAG, the other lipid second messenger, activates the enzyme protein kinase C, which then phosphorylates a large number of proteins, leading to the cell's response. In addition to these effects, the lipid portion of DAG is arachidonic acid, which is the precursor for the prostaglandins and other local hormones that cause multiple effects in tissues throughout the body. And calcium calmodulin second messenger system. Another second messenger system operates in response to the early entry of calcium ions into the cell. Calcium entry may be initiated by first method, changes in membrane potential that open calcium channels, to hormone interacting with membrane receptors that open calcium channels, and upon entering a cell, calcium ions bind with the protein calmodulin. The calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinase. Activation of calmodulin dependent protein kinase causes phosphorylation, activation or inhibition of proteins involved in the cell's response of the hormone. For example, one specific function of calmodulin is to activate myosin light chain kinase, which acts directly on the myosin of smooth muscle to cause smooth muscle contraction. And the normal calcium ion concentration in most cells of the body is 10 raised to minus 8 to 10 raised to minus 7 mole per liter, which is not enough to activate the calmodulin system. But when the calcium ion concentration rises to 10 raised to minus 6 to 10 raised to minus 5 mole per liter, enough binding occurs to cause all the intracellular actions of calmodulin. Then hormones that act mainly on the genetic mechanisms of the cell. Steroid hormones increase protein synthesis. Another means by which hormones act, especially the steroid hormones, is to cause synthesis of proteins in the target cells. These proteins then function as enzymes, transport proteins or structural proteins, which in turn provide other functions of the cells. The sequence of events in steroid function is especially the following. The steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds with a specific receptor protein. The combined receptor protein hormone then diffuses into or it's transported into the nucleus. The combination binds at specific points on the DNA strands in the chromosome, which activate the transcription process of specific genes to form mRNA. The mRNA diffuses into the cytoplasm, where it promotes the translation process at the ribosome to form new proteins. To give an example, aldosterone, one of the hormones secreted by the adrenal cortex, enters the cytoplasm of renal tubular cells, which contain a specific receptor protein often called the mineralocorticoid receptor. After about 45 minutes, proteins begin to appear in the renal tubular cells and promote sodium reabsorption from the tubules and potassium concentration into the tubules. Thus, the full action of the steroid hormone is characteristically delayed for at least 45 minutes, up to several hours or even days. This action is in marked contrast to the rapid actions of some of the peptide and amino acid derived hormones such as vasopressin and norepinephrine. So, thyroid hormones increase gene transcription in the cell nucleus. The thyroid hormones thyroxine and triiodothyronine cause increased transcription by specific genes in the nucleus. To accomplish this increased transcription, these hormones first bind directly with receptor proteins in the nucleus. These receptors are activated transcription factors located within the chromosomal complex, and they control the function of the gene promoters. The following two features of thyroid hormone function in the nucleus are important. They activate the genetic mechanism for the formation of many types of intracellular proteins, probably 100 or more. Many of these intracellular proteins are enzymes that promote enhanced intracellular metabolic activity in virtually all cells of the body. Once bound to the intracellular receptors, the thyroid hormones can continue to express their control functions for days or even weeks. The measurement of hormone concentrations in the blood. Most hormones are present in the blood in extremely minute quantities. Some concentrations are as low as 1 billionth of a milligram, 1 pg per milliliter. Therefore, it was difficult to measure these concentrations by the usual chemical means. However, an extremely sensitive method that was developed about 50 years ago revolutionized the measurement of hormones, their precursors, and their metabolic end products. This method is called radioimmunoassay. More recently, additional methods such as enzyme-linked immunosorbent assay or ELISA tests have been developed for accurate, high-throughput measurements of hormones. Radioimmunoassay, we will be explaining that again. So, the method of performing radioimmunoassay is as follows. First, an antibody that is highly specific for the hormone to be measured is produced. Second, a small quantity of this antibody is mixed with a quantity of fluid from the animal containing the hormone to be measured and mixed simultaneously with an appropriate amount of purified standard hormone that has been tagged with the radioactive isotope. However, one specific condition must be met. There must be too little antibody to bind completely both the radioactively tagged hormone and the hormone in the fluid to be assayed. Therefore, the natural hormone is the assay fluid and the radioactive standard hormone competes for the binding sites of the antibody. In the process of competing, the quantity of each of these two hormones, the natural and the radioactive that binds, is proportional to its concentration in the assay fluid. Third, after binding has reached equilibrium, the antibody hormone complex is separated from the remainder of the solution and the quantity of radioactive hormone bound in this complex is measured by radioactive counting techniques. If a large amount of radioactive hormone has bound with the antibody, it is clear that there was only a small amount of natural hormone to compete with the radioactive hormone, and therefore the concentration of the natural hormone in assayed fluid was small, and the converse is also true. Fourth, to make the assay highly quantitative, the radioimmunoassay procedure is also performed for standard solutions of untagged hormone at several concentration levels. Then a standard curve is plotted. By comparing the radioactive counts recorded from the unknown assay procedures with standard curve, one can determine within an error of 10 to 15% of the concentration of the hormone in the unknown assayed fluid. As little as billions or even trillions of a gram of hormone can often be assayed in this way. The next method we just mentioned, enzyme-linked immunosorbent assay. ELISAs can be used to measure almost any protein, including hormones. This test combines the specificity of antibodies with the sensitivity of simple enzyme assays. Basic elements of this method, which is often performed on plastic plates that each have 96 small wells. Each well is coated with an antibody AB1 that is specific for the hormone being assayed. Samples or standards are added to each of the wells, followed by a second antibody AB2 that is also specific for the hormone but binds to a different site of the hormone molecule. A third antibody AB3 that is added recognizes AB2 and is coupled to an enzyme that converts a suitable substrate to a product that can be easily detected by colorimetric or fluorescent optical methods. Because each molecule of enzyme catalyzes the formation of many thousands of product molecules, even small amounts of hormone molecules can be detected. In contrast to competitive radioimmunoassay methods, ELISA methods use excess antibodies so that all hormone molecules are captured in antibody-hormone complexes. Therefore, the amount of hormone present in the sample or in the standard is proportional to the amount of product formed. The ELISA method has become widely used in clinical and research laboratories because it does not use radioactive isotopes. Much of the assay can be automated using 96 well plates, and it has proved to be a cost-effective and accurate method for assessing hormone levels.